Today we're going to talk about positive and negative selection of thymocytes. Thymocytes occurs in the thymus, and there are two regions that these cells have to go through, which is the cortex, and that's where the positive selection takes place, and the medulla, that's where the negative selection takes place. In the cortex, you get positive selection because it requires a positive interaction between the T cell and a receptor within the system. Let me show you a picture here. Okay. As you can see here on the picture, the top portion is positive selection and the bottom portion is negative selection. So in the cortex, it's an interaction, requires a positive interaction between the T cell and a receptor within the system. This T cell can recognize and interact with self-MHC. If you get some T cells, specifically double positive T cells, that can interact with MHC on the surface of epithelial cells, and they interact too strongly, they will recognize the MHC, but they recognize it in a way that they might overreact, you can say. So they don't respond positively to the reaction. Then they have to be killed off. Those cells will have to be killed off because we do not want them to attack our cells. If the T cells do not interact at all with the MHC peptide, then they die off. It's a positive selection because it is positively interacting with the MHCs. And you have two types of MHC molecules you can see right here. You have the CD4 T cell, and you have a CD8. So the interacting MHCs can produce CD4s, which is a type of MHC that they interact with, and they can produce CD8s, and this is based on the type of MHC they interact with. So based on the type of MHC, it depends on what MHC is able to interact. If it's able to interact with MHC1, the cell becomes CD8 T cell. So if it interacts with MHC1, the cell will become CD8, which is this here. And if it interacts with MHC class molecule 2, then the cell becomes CD4 T cell, which is also known as a helper T cell. And the CD8 also is known as a cytotoxic T cell. And cytotoxic T cells, they're able to remove viruses. They're able to remove virus-infected cells. And they also can remove neoplastic cells. The formation of helper T cells, there are two types. There's a T helper 1 cell, and there's also a T helper 2 cells. T helper 1 cells, they are created with the influence of interleukin 12. So interleukin 12 is based upon T helper 1 cells, and T helper 2 cells are based on the influence of interleukin 4. And those are the two types of cells that will form if reacting with the class 1 MHC class 1 molecule. So then it moves down. This is all of this here is the cortex. So here we're moving down to the medulla right here, as you can see, the medulla. In the medulla is where negative selection takes place. Negative selection takes place because the CD4s and CD8s will move into the medulla and then interact with more thymic epithelial cells, so they'll interact with other cells. This interaction is negatively selected if it reacts too strongly. So I kind of described it a little bit when I was talking about the cortex and the positive selection, but I'm going to go into more detail. So actually, when it reacts too strongly, that's when you get a negative selection. And the interaction is negatively selected if it interacts too strongly with the antigens that are being expressed. So the mature T lymphocytes will then immigrate out and can enter the bloodstream. So once the CD4 or CD8s pass through the cortex, through the medulla, it is then immigrated out and enters the bloodstream. Negative selection, after the cells have differentiated into CD8 T cells and CD4 T cells, it will go to the medulla. Negative selection checks how capable it checks how capable the new T cells will be able to bind to self MHC peptides. So, if the interaction is too strong, which I already stated, the formation of self-reactive T cells that are capable of inducing an autoimmune disease. So that's where, one of these things can happen far as like what goes on in our body. So when you get a negative selection or negative reaction, and it reacts too strongly, it produces autoimmune diseases, and that's why we want the interaction to be just right. That's why we need it to be just right, so that we can get the right response. Once it passes through both of these processes, the CD4 and CD8 T cells now becomes mature helper T cells and cytotoxic T cells. So they start off as immature cells, and once they pass through the cortex and the medulla, they become mature cells. And also these cells that are negatively selected, they can go through a process called apoptosis, and apoptosis is basically programmed cell death. Okay, so that's how the cells die off if they don't interact correctly, and that's how they'll die off.